Now the tertiary structure involves other factors like hydrogen bonding, ionic bonding, the hydrophobic effect, because like it has like stuff. It's like hydrophobic, you know what I mean, you know, and also because so it gets like all crumpled up. Now the quaternary structure involves like 2 pages combining, so you get. So
Here's the thing.
It's strong. It's never
I'm
wrong.
gonna switch out. I'm gonna say.
It's strong. How about a covalent bond?
Well, with your question about covalent bond, well, basically what happening is this: you have 2 nonmetal atoms. Okay, let's say for example, just to make things simple. Let's say these are 2 hydrogen atoms. Just make things simple. Now, how many valence electrons does each one have?
One.
1 valence. That's correct. And I'll use the red dot for the valence electrons. Okay. And then what happened is, when these 2 bond together, this is how they bond. Well, since they both the same atom, for example, neither one will want to lose or gain the other valence electrons. So you have these 2 atoms, they wind up sharing their valence electrons. So this valence electrons will be shared with this paper. This atom will also share its valence electrons with this paper. So as a result, you have these 2 atoms here, a hydrogen, they're sharing their valence electrons. Now, you say, well, how does that allow them to stay together? Well, you notice in the atom, isn't a center positive? Isn't a center atom positive? So I'll make a note, excuse me here, I'll just erase this one. The center here being positive, the center here being positive. Now we notice the positive being pulled toward this negative center of these electrons, and this positive being pulled to this negative center. So you have these 2 atoms being pulled to each other. As a result, that's how they bond. And that bond right there, we call this
Covalent bond.
covalent bond. Now, let's look at this term. Co, you learned from your English class, means together, right? Sharing together. And what do you think valent refer to? Valence electron. So it's a bond due to sharing of valence electrons.
Golly gee, Mr. Strong. How did you know that's right?
It's a tron, it's never wrong. Okay.
Now, what does this look like to you?
A tree?
Pastrami sandwich.
No, you guys are wrong. Those are genes. And where are genes found? Yeah, that's right. The 4th type of organic molecule, nucleic acids. It's DNA.
Yeah.
Okay, all the genetic information of a cell is stored in nucleic acids. Now, an example of it is DNA. DNA is a polymer of nucleotides. Now, these nucleotides consist of 3 parts: a 5-carbon sugar, a phosphate group, and a DNA and a nitrogen base. Now, there are 4 types of nitrogen bases. Well, 2 types actually. There's purines and pyrimidines. So now, just for your benefit, for an analogy, we have pens and pen caps. Now we can imagine that these pens are purines and these pen caps are pyrimidines. Now here we have a purine called adenine and another purine called guanine. Now for pyrimidines, we have, for this one, thymine and cytosine, see, pyrimidines. As you can see, purines bind with pyrimidines, but only in a specific order. Now, thymine can only bind with adenine, see, it doesn't fit with guanine. It just doesn't work, yeah. And then, cytosine can only bind with the guanine, see? It's just perfect, it doesn't fit in the other one, it gets all messed up. Now, there are several differences between DNA and RNA. First of all, sugar is ribose and not deoxyribose, and the thymine pyrimidine is replaced with the uracil. See, uracil, see, yeah, uracil, so it's replaced with uracil, and it binds to the adenine itself. Yeah, and also RNA is usually single-stranded while DNA is double-stranded, but you don't need to know that, just a bunch of crap. Yeah, bunch of crap. Now, introducing the new superhero of the town, ATP Boy! Look at him exert all that ATP. Ooh, 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 ATP. Also known as adenosine triphosphate, is a common source of energy in living systems and stuff. In order for it to provide energy, it's converted to ADP, adenosine diphosphate. It loses a phosphate group and everything on it and just seems to provide energy somehow. And then in order to re-input the energy into ATP, you have to phosphorylate it, which means adding a phosphate group to the ADP, and you get like your ATP back. Yeah, ATP. Extreme. When you start recording.
Go.
Oh hey folks, I didn't see you there. So now folks, do you know what an enzyme is? Okay, anyway folks, enzymes are globular proteins that catalyze reactions. They work by having a substrate molecule bind to an active site. Now this induced fit model is very substrate specific. You know, not just any substrate will work on any enzyme. So now folks, just imagine that all these cars are enzymes and this one key is a substrate. Now which enzyme will bind to the substrate? Hey, what are you doing in a car? Okay, folks, that was enzymes. I'm a little enzyme, small and substrate specific. Here is our binding site, and that's all we have. When it gets too acidic or steamed up, we denature and rock and roll! Do rocka! Okay everyone, now we'll go over some cofactors. Cofactors, they like assist enzymes and like, yeah, and doing their duty and stuff like that. And there are two types. There are coenzymes, which are organic cofactors. Some examples are like vitamins and stuff. And then you got your inorganic like cofactors, which are like some examples like metal and iron and stuff like that. Yeah.
Time for the activation energy extravaganza.
Every reaction needs energy to work. Imagine this tennis ball is a chemical reaction and I hit this over but not get enough energy to it. See? It didn't go past the net, but when you give enough energy to it, like this.
Hold on, you fucker.
Phosphorylate.
Sorry.